Hello everyone, this is the Imaris guy. Welcome to the next video in this series. In this video we're going to cover the extension called synaptic pairing. Mainly this extension is going to be used to further the advancement of the colocalization of two sets of spots within a 3D volume. As with all of my extensions, these extensions can be found on my GitHub page shown here. This particular one is version 9, Synaptic Pairing 9 is the one that we're going to go over today. You ever find yourself in an image that looks like this, multiple labels, 2 labels, possibly even 3 labels at sometimes, where you want to try to coordinate and colocalize the different spots, whether you have a presynaptic spot or a postsynaptic spot, but you want to be able to kind of get some relationships between the two. Now, Imaris has a lot of built-in tools to not only segment these objects, whether it be surfaces or spots objects, but we do have a bunch of tools and ways to quantify the number of red spots that are colocalized with green. So if we zoom into here and we look at all these structures, we go into our slicer mode, we can come up here and we can look in our slice mode. Oh yeah, there's a red spot and a green spot, those are colocalized. There's a red spot and a green spot, those are colocalized. However, in some cases, and in a case like this particular data set, some of these spots have multiple colocalizations within the structures. For example, you could potentially have 2 spots that are colocalized. There's 2 red spots possibly colocalized with 1 green spot. There might be 3 red spots colocalized with green, or 2 green spots colocalized with red. This type of analysis is something that's really unique in and being able to identify, different pairs and things like that within the structure is something that I wanted to kind of investigate and see if we can't make an extension to try to kind of enhance that type of analysis. So not just looking at the number of red spots colocalized with green spots, which is very useful and very powerful, but also kind of take it to another level and try to see if there's a little bit other structure within this type of analysis after we do the segmentation to try to kind of enhance and further the analysis of the synaptic organization, or the pre-synaptic, post-synaptic, or it could be any kind of 2 puncta that are 2 different structures that you're looking for those relationships between each other. And so synaptic puncta, whether they're PSD95 or V-glut or glutamate receptors, or any kind of structure that kind of labels in this punctate manner, is going to really kind of benefit within this particular extension. So if we do have a data set like this, again, this is a 2D slice. You can look at the structure. It's a pretty big stack of data set. There's a lot of spots within this data set, and it's really good. And there's some a little out of fluorescent stuff here from the puncta. That's pretty typical with a lot of antibody stains out there that are labeling like this. But we're going to focus on the bright puncta structures. Now, there's 2 ways to label puncta inside of Imaris. One is with the very simple and fast spot detector, and the other is a surface rendering. I'm going to focus right now in the beginning talking about spot detection and then using the extension on spots to show you what the results of that analysis would be. So let's get into that. Now, I already have the spots detected. I'm not going to get into the creation of the spots within the process, but I went through and I have red spots and I have green spots. And I used about the parameters I used, just so you're aware. I used a spot size of 0.4 microns, and again, if you go in here and measure the diameter of some of these red spots and green spots, if I come up here and measure that, it's about, if you look over here, it's about 4 microns in diameter. So that's where I came up with the 4 microns, and I think I used 4 for both of them. Even though the green is a little bit bigger than the red, I think I ended up using the same spot size for the red and the green channel. And again, if we turn off this channel, we look at our red spots, look at our green spots, you get something that looks like this. And again, we have thousands of spots here. So again, if we look at our data sets, we have 27,000 red spots, and we have 82,000 green spots. So many, many, many more green spots. But the segmentation looks pretty good. If you look at one or the other, let's just look at the red spots here. If I zoom in here, turn on just the red channel, we can zoom in the slicer mode, and you can see you're going to have 1 spot as you go up and down through the stack for every puncta within the structure, and again labeled right smack dab in the center of those puncta. So when we go and do our particular extension, it is going to be measuring distances between the center of those spots. So what we're now going to do is going to launch the Imaris extension Synaptic Pairing number 9. And to do that, I'm going to click on the green object here. Click on the gear tab is where I have all my extensions. I'm going to look for Synaptic Pairing number 9. That's this one down here. Again, when you launch this, it will create a little dialog box that will pop up. It'll ask you to choose the spots objects that you want. it'll look something like this. There's a couple different options in here. You can choose whatever objects. It should list all of the spots objects that are here. And it'll have a distance threshold. Now, this process is based on distance, center of one spot to the center of the other spot. So you want to choose the spot size here that's relevant to the objects you're trying to detect, and the size of the spots you use to detect those objects. In this case, I used a 0.4 micron spot size, so we want to probably use about 0.4 microns here. So I'm going to set that to 0.4 in the process. Now, if you do come down here and change this thing, it does look like these things get unchecked, so just make sure we turn those guys back on, and then you're just going to click the Analyze Coloc button. Now this is going to be real time. It only takes about a minute or so to launch. I'll bring this over here. There is a progress bar that will pop up and run through this process of calculating these spots. Now what this is doing and what the processing is doing is looking all of the spots, all 18,000 of these spots, 15,000 or whatever the other spots or whatever, and they're calculating the distance between them, calculating them, and then it's going to create a bunch of labels. So we're going to go through that in a few seconds here when this is done. And you're going to see when it's done, it will rename the green and the red spots with a colocalized term. When it's all done, you'll see that in a second. There we go. So now you have a red spot and a green spot colocalized, right? So each one of these spots, it has all the spots that are colocalized. So the way I've organize this, I'm going to turn off the red spots. Actually, you know what? I'm going to turn off. So here's just mainly just a matter of kind of visualization of the data. So I'm going to turn off the labels for the red spots. So all the red spots will look red. I'm going to go to my green spots. And so inside the green spots, if you go on the label tab here, you're going to see a whole bunch of brand new labels that get generated here. You have the non-colocalized spots, which are the spots that are not within the threshold that we set for the distance measurement. We have the ones that are singles, one spot, one red spot, one green spot. We have doublets, which are basically two green spots and one red spot. We have doublet special, which I've identified something that has a doublet and a singlet together. So I just kind of called it this doublet special because it's not quite a doublet, it's not a singlet, it's kind of the combination. And I'll show you an example of that in a second. And then there are triplets. Basically, there are 3 green spots surrounding one red spot. Okay, so let's go ahead and try to find some of these different examples. So I'm going to go into the color tab here. The color tab here has a bunch of different options to kind of make the visualization here a little bit cleaner. Right now we have what's called the labels overriding the base color. The base color is green, so it's overriding those colors with all these colors. So every single spot has a label, whether it's non-colocalized or colocalized, doublet, singlet, whatever. I'm going to switch this to only show the objects that have a label or visible event. So when I do that, I have this checkbox here. So now I can come in here and I can turn off all the non-colocalized spots. I can just turn them off and we don't have them visible. Now what's present is just the spots that are colocalized. The ones that are yellow here are my singlets. So if I zoom in to a couple of these guys here, these 2 spots down there, colocalized. These 2 spots, colocalized within that 0.4 micron threshold distance. Now, we also have some doublets here. So I'm going to turn off the singlets. We're going to try to find some doublets. So the doublets, let's see if we can find a couple. Those are going to be the purple, dark purple guys. And you can see here, there are 2 spots here associated with 1 red spot. That would be considered a doublet. Those 2 green spots, 1 red spots. Then we also have doublet specials. Now again, what were the doublet specials? Doublet specials basically are a doublet and a singlet kind of together. So let's see if we can find some of these guys here. These are going to be orange. So let's do this guy right here is an example of a doublet special. So basically what it means is that this here, these 2 orange guys are a doublet with that guy, but it's a singlet. This guy's a singlet with that guy. So that's what I call the doublet specials to identify this kind of scenario as well. And then we also have the triplets. So I'm going to turn off that guy. I'm going to try to find a triplet here. See if we have one in here. There's a couple. So here's one in here. Basically, 3 green spots associated with 1 red spot. So that's essentially what the extension does. It kind of identifies these doublets, identifies these triplets. Now, and it does this for both channels, right? So these are the green, this is the triplets associated with the green spots associated with the red. Now, if we flip this across, again, I'm going to turn off this here. I'm going to make all the green spots green, so turn off the labels. I'm going to go to the red channel. I'm going to go to here. I'm going to override the colors here and only show the labels. Again, get rid of the non-colocalized. the green, yeah, the non-colocalized there. So all the, oh, sorry, wait. Oh yeah, there's just many more green spots, I guess. It just looks weird. Okay, so the idea here is you have, again, your singles are the purple guys. One spot, you kind of rotate it around, you can kind of see one spot there, green spot there, right? One red spot, one green spot. Turn off the singles, let's find the doublets, same deal, right? You come here, try to find 2 purple cells that are associated with 1 green spot within the distance threshold that we set, and so forth and so on. So all these objects, all these labels are made for each one of these objects. I believe the particular extension goes up to quadruplets, so if there are 4 red spots with 1 green spot, it will come up with those as well. The final summary of this extension rests in the quantification of the labels of the different categories of the colocalization that we've identified. Colocalized and the singles are just pure numbers, right? So there's 16,327 non-colocalized red spots. We're in the red statistic here. Under the singles, there's 10,491 singles. 1 red spot, 1 green spot. For the doublets, now remember, each red spot is paired up. So this number is doubled. So each red spot, when you're looking at a doublet, in this case these blue guys, it's counting each one of these spots individually. So 1, 2. So if you want to get the total number of doublets within the entire data set, you just have to divide this by 2. You get 432. Doublet special, you'll get 54, and then the triplets, same thing. Here you divide it by 3, and you'd say there's 5 triplets. So any of the white ones here, there's 5 sets of triplets. There's 432 sets of doublets within this particular example. So that's where these numbers come from. At some point, maybe I'll kind of unify it up and get an overall statistic in there where we can put it into the overall tab here and get overall number of doublets, not just the number of spots within each structure. That might be a little bit clearer. Maybe I'll add something like that in the future. Thank you for listening to the tutorial session on the Synaptic Pairing extension. I hope you enjoyed it. This was the Imaris guy. Until next time, thank you.